Perfect. All right. So hi, everyone, and thanks a lot for attending this month's NeuroMeetup talk. So today, our first speaker is going to be Natalia Valle-Tamayo. So Natalia received a degree in biomedical science at the University of Barcelona in 2019. And during the final year of a degree, she did an internship at the University of Leicester in the UK in the Neuroscience, Psychology and Behavior Laboratory, where she carried out a final degree project on the study of the early stages of amyotrophic lateral sclerosis. In 2020, she obtained a master's degree in translational biomedical research at the Vall d'Hebron Research Institute. And during the same year, she joined the Sant Pau Memory Unit in Barcelona, precisely to study neurodegeneration in inflammatory biomarkers in Alzheimer's disease and in Down syndrome. So currently, she's coursing a PhD in neuroscience at the Autonomous University of Barcelona, where she focuses mainly on the use of extracellular vesicles as a new platform for the study of biomarkers in Alzheimer's disease. So the title of the talk is going to be Exploring Biomarkers of Basal Forebrain Cholinergic Degeneration in Neuronal Extracellular Vesicles. So Natalia, the floor is yours. Natalia, we cannot hear you for the moment. No, we still can't hear you.
Now?
Now we can, yes.
Okay, sorry. Thank you very much for your presentations. I'm very grateful for being here. So, well, today I'm going to talk about my research in exploring biomarkers of basal forebrain cholinergic degeneration in Down syndrome. So as goals of this session today, I will go through the cholinergic deficits in Alzheimer's disease and Down syndrome, why we have interest in pro-NGF as a biomarker, and extracellular vesicles as non-invasive tools for the study of biomarkers. So Alzheimer's disease is the most common neurodegenerative disease affecting about 50 million people in all the world. The most characteristic pathological features of this disease are brain atrophy, amyloid plaques, and neurofibrillary tangles. It also constitutes the most common form of dementia in the elderly. However, there are other populations that can suffer it, such as autosomic dominant forms and people with Down syndrome. Especially Down syndrome is a population that have a triplication of the chromosome 21, which harbors the APP gene that encodes for the amyloid precursor protein that produces the A-beta amyloid. Therefore, people with Down syndrome accumulate these lesions during their lives, and more than the 90% develop dementia in the adulthood. Then, what's the relationship between AD and the cholinergic damage? AD causes cognitive impairment and loss of the memory, and these deficits are closely linked to cholinergic deficits. For instance, some neuropathological studies performed in the 70s identified a loss of activity and decrease in the levels of enzymes involved in the synthesis of the acetylcholine in the brains of patients with AD, such as the choline acetylase. It was also reported that basal forebrain cholinergic neurons were atrophic and small in number in brains affected by AD. In addition, subsequent studies demonstrated that such loss of activity of these enzymes were associated with poorer cognition. So these cholinergic neurons are important since they play a central role in cognitive functions like memory, attention, and learning, functions that are also impaired in AD and Down syndrome. A peculiarity of these neurons is that although during neural development, all neurons depend on growth factors for survival, only these basal forebrain neurons retain this dependency in the adult brain. Therefore, they need a continuous supply of the NGF, the nerve growth factor, for survival, phenotypic maintenance, and synaptic integrity. And the biological action of the NGF is mediated by the high affinity receptor TRK with tyrosine kinase activity. So we have that on the one hand, there is a loss of quantity and function of the choline acetylase activity, and that there is an atrophy of the basal forebrain cholinergic neurons that in turn are NGF dependent. And however, in the other hand, we have that there are normal NGF mRNA levels in the AD brains, and the levels of the NGF precursor protein pro-NGF are increased. Therefore, it is paradoxical that a cholinergic atrophy of the NGF-dependent neurons coexists with this elevated supply of the NGF precursor molecule. And the question here would be, why is trophic support not preserved in AD? So what is going on with this NGF in AD and Down syndrome brains then? Before I met her, my thesis supervisor, Florencia, was part of the Claudio Cuello labs in Canada, and this group elucidated the NGF metabolic pathway and its dysregulation in AD and Down syndrome. So basically, the impairment is based on the fact that the pro-NGF accumulates because the enzymatic mechanisms responsible for its maturation to NGF are affected in AD. And this compromises the availability of the mature NGF, and that also explains the selective vulnerability of these cholinergic neurons in this disease. Then the question was if we can measure pro-NGF and if these changes in pro-NGF will also be reflected in accessible biofluids such as blood. So they started the first study to measure pro-NGF in plasma of people of Down syndrome, and they saw increased levels in Down syndrome and Down syndrome with AD compared to healthy controls. And in addition, they also show that these increased levels correlated with cognitive impairment. At that point, they still had no CSF, and this is where the second study was born. And later in the second study, they had also CSF, and then Julita and colleagues measured pro-NGF in both plasma and CSF samples from healthy controls, Down syndrome without symptomatic AD, and Down syndrome with symptomatic AD. This by Western blot, and to see if these changes could be also reflected in these biofluids. So as reflected in the graphs, higher levels of the pro-NGF could be detected in both biofluids. However, it was only in CSF that they were able to detect significantly increased levels of pro-NGF in symptomatic compared to asymptomatic individuals. Then looking to the RAC curve analysis, only CSF pro-NGF has a good diagnostic performance to distinguish symptomatic AD and comparable to core AD biomarkers. Although these results support the hypothesis that CSF pro-NGF could have potential as biomarker in brain degeneration associated with AD, at that point there were still difficulties to be resolved, because the advantages of CSF analysis are unquestionable, since it's the liquid that is in direct contact with the brain, having an exceptional reflection of the brain's neurochemistry. However, despite involving minimal risks, lumbar puncture is still perceived as invasive and need specialized trained healthcare professionals, which makes it complicated for longitudinal studies and follow-ups. And that is why there has been strong interest in finding blood-based biomarkers. In relation to that, for the past few years, the analysis of cell-specific extracellular vesicles in biofluids has revolutionized the field of biomarkers. EVs are lipid-delimited nanoparticles naturally secreted by all types of cells and play important roles in cell communication, metabolic elimination, and transfer of pathogens to other cells. And as they are delimited by a lipid bilayer, the intravesicular contacts are protected from degradation. And as they are present in all biofluids, they can easily be analyzed by liquid biopsy in a non-invasive manner. For these reasons, EVs will provide us with the possibility to interrogate cerebral targets in plasma protected within these brain-derived EVs. And that's how the project I started working on with Florencia was born in 2020. She was granted the Jerome Legend Foundation, and this grant also allowed me to join the group as a PhD student. The objectives of that project were first to set up a methodology for the enrichment of neuron-derived EVs, referred as NDEPs, and to analyze this pro-NGF in NDEPs along the AD continuum in Down syndrome and healthy controls. The method that we finally optimized consisted on a first isolation of the EVs by a precipitation-based method and an immunoprecipitation with L1-CAM antibody to select these neuron-derived EVs. And well, first of all, we optimized this method and performed some validation checks. We verified the size and morphology of EVs by cryo-electron microscopy and nanoparticle tracking analysis. Nanoparticle tracking analysis, represented by this graph, is a technique that allows us to quantify both size and concentration of EVs. We also verified the presence of vesicular markers by Western blot. We performed a lipoprotein contamination test by ELISA, and that demonstrated that we could achieve a quite pure NDEP fraction. And in addition, we confirmed the enrichment of neuronal markers such as L1-CAM, neurofilament, and p-tau1 by ELISA and Simoa. Therefore, we indeed achieved the enrichment of these neuronal EVs. Well, regarding the cohort we use, I have to say that we are very lucky that our group have the largest and best characterized population cohort of individuals with Down syndrome worldwide, called DAPNI. DAPNI refers to Down Alzheimer Barcelona Neuroimaging Initiative, and this cohort is part of a health plan that offer annual neurological and neuropsychological assessment for adults older than 18 with Down syndrome in Catalonia. And these professionals involved have created a very large biomarker cohort consisting of biofluid and biomarker imaging biomarkers. So from this cohort, we selected 127 participants, 21 healthy controls, 41 people with Down syndrome with asymptomatic AD, and 65 people with Down syndrome with symptomatic AD. That involved both prodromal and dementia ones. And we isolated neuron-derived EVs from the plasma of these individuals and measured pro-NGF to see changes in the AD continuum. So moving to results, as shown in this graph, pro-NGF was significantly elevated in NDEPs from Down syndrome compared with healthy controls, and we observed a trend suggesting an increase in pro-NGF in symptomatic than AD in Down syndrome. If we split the Down syndrome groups, we could see that the increase of pro-NGF is driven by the demented Down syndrome individuals, as you can see here. And on the other hand, it's well known that in Down syndrome, there are endosomal anomalies and a greater secretion of EVs compared to diploid individuals. Therefore, we quantify the vesicular marker CD81 in the same NDEP samples that we have previously quantified pro-NGF. And the analysis demonstrated that Down syndrome individuals saw higher CD81 levels compared to controls. So we use these results to normalize the pro-NGF levels. Once we did the normalization, we were able to detect significant increased levels of the ratio of pro-NGF CD81 in symptomatic Down syndrome individuals compared with the asymptomatic. And if we split the Down syndrome groups, we saw no significant changes between the prodromal and the metric ones, but we did saw differences between asymptomatic and prodromal. And we are now exploring the relationship of these pro-NGF levels with other variables such as age, sex, ApoE, genotype, and etc. Finally, as a summary of all I have been through today, I would like to point out that basal 4 brain cholinergic neurons have a strong dependency on NGF to maintain a healthy phenotype even in the adult brain, that pro-NGF can be measured in free plasma CSF and in neuronal EVs, and that pro-NGF in CSF and in neuronal EVs increases along the AD continuum in AD. In Down syndrome, sorry. And well, just to finalize, I would like to acknowledge all my team from the San Pau and especially my supervisor, Francia. Thank you.
Thanks a lot, Natalia. Thanks a lot. That was very interesting. So please, to members of the audience, don't hesitate to ask questions, to write them down directly into the chat so that I can ask them directly to the speaker. For the moment, we don't have any questions. I'm just going to wait just a little moment for people in the audience who might be willing to ask a question.
Okay.
Maybe me, Natalia. Can you hear me?
Yeah.
Okay. So I have a few questions just out of curiosity. So
Okay.
I was wondering, you said at the beginning that these cholinergic deficits occur in several areas of the brain. And I was wondering if they all occur at the same time or if there is an area of a starting point and then you observe it.
Yeah,
in a
I
time
don't
course.
really know if it's like, if it starts specifically in the nucleus basalis and then spread. I think it's the whole basal 4 brain, like nucleus basalis and mediastal nucleus, but I can tell you by sure.
Okay. Yeah, then I was wondering, you mentioned that there is a basal level of NGF and pro-NGF as well in plasma, CSF, in all these biofluids, and that cholinergic neurons are dependent on this NGF trophism.
Yeah.
And that you extracted these vesicles and that you found pro-NGF only. I was wondering, do you have any level of NGF as well, meaning that the enzyme for the cleavage or the maturation of the NGF is just downregulated or is completely depleted?
I don't think it's completely depleted. Before we bought the ELISA kit, we checked that there was no cross-reactivity to ensure that we were only measuring pro-NGF and not NGF. But we are doing in parallel a study that with the Olink platform. No, I don't know if you have done, well, it's an ultra-sensitive technique. And we have been showing that in Down syndrome, beta NGF is not detected in plasma nor CSF in Down syndrome.
Okay.
But it's preliminary results. I cannot tell it by sure, but it was a full cohort of about 100 people, and NGF was not detectable in Down syndrome, nor plasma nor CSF.
Okay. Can I ask you as well? I was also studying the NGF signaling, but in pain mechanisms in the peripheral nervous system. Basically, NGF is present in the inflammatory milieu, and so it's also binding constantly TrkA receptors and then activating the nociceptors. So I was wondering, instead in cholinergic neurons, what is the signaling, the downstream mechanism? What does it do? How does it regulate the trophies?
You mean like the pathway of the gene?
I mean, how would you say it's strictly NGF dependent and not any other growth factor, for example?
Okay, I haven't studied that, but Florencia was doing some experiments in Canada, and they pick up some brain and they depleted the NGF. They blocked the receptor of the NGF, and they see the atrophy and all of the generation I have been talking about. So I suppose that this NGF is needed and cannot be supplied with other neurotrophin.
Okay. Okay. Maybe just one last question.
Okay.
So when you compare Alzheimer's disease patients with the Down syndrome patients instead, there is, let's say, a time dependency that is quite different, because people with Down syndrome are born like this, whereas you get Alzheimer like in time. I was wondering how you correlate this time difference.
But I didn't select people with Down syndrome and separately people with Alzheimer's disease.
Okay.
All the cohort was Down syndrome, and the key was to use this cohort, knowing that it's a population that will by sure develop Alzheimer's disease.
Okay.
So we code it in different stages, like asymptomatic, all checked by biomarkers and all, and check asymptomatic, prodromal, and demented ones. And the statistic, we compile both prodromal and demented asymptomatic, but they were not separately AD or Down syndrome, were all Down syndrome studies.
Okay, thank you. That was really informative.
Yeah, thanks a lot. I suggest we switch to Federica's talk, if that's okay for you. So Federica, I just let you share your slides.
You always have to tell me how it looks.
Yeah, you go in the upper right corner, lower right corner.
See. Display settings again, and like this.
Yes, exactly. Perfect. It's working nicely. All right. So just a quick introduction for the second talk. Federica studied biology at the University of La Sapienza in Rome and specialized in neuroscience during her master's program. For a master thesis at the EMBL Neuroscience Unit, she focused on nerve growth factor signaling on nociceptors and was involved in developing a targeted treatment for chronic inflammatory pain. She then moved to Bern in Switzerland, where she is currently doing a PhD in neuroscience in the lab of Professor Thomas Nevian, studying chronic pain. And the title of the talk is going to be Physiology and Morphology of Layer 5 Pyramidal Neurons of Anterior Cingulate Cortex in Inflammatory Pain. And Federica, the floor is yours, but we still see both screens.
Both screens.
Yeah, you have to go to display settings and swap.
I'm sorry, exactly. I continue mistaking. Okay. So thank you for the introduction, Camille. So my talk today is called Layer 5 Pyramidal Neurons of the Anterior Cingulate Cortex in Inflammatory Pain. And I hope to give you a brief introduction of what my PhD project has been so far. So we know that the anterior cingulate cortex is a substructure of the prefrontal area that is located in a very peculiar place that is surrounding the initial part of the corpus callosum, and that this area is and its connections is highly preserved across mammals. It is also very well interconnected with other regions in the cortex, like the medial orbital cortex, ventral orbital cortex, but also the thalamus, basal forebrain, and indeed it plays a central role in various functions related to behavioral flexibility, such as error detection, reward, aversion, and the pain processing, which is important for my project. In fact, if we follow the nociceptive pathway from the periphery through the spinal cord, we can see that the nociceptive information is then received in the brain from areas that are involved in the sensory modulation of pain, like, for example, the thalamus and the somatosensory cortices, and regions involved in mediating the emotional states of pain, like the amygdala or the insular cortex. We see in the middle that the ACC receive inputs from various cortical and subcortical structures, and what happens in the ACC is that the neurons in the deep layers that constitute the major output population of this brain area send their projections directly or indirectly then down to the dorsal horn of the spinal cord. So this is a nice example of top-down circuit that allows cortical neurons to directly modulate the sensory input into the central nervous system. But differently from what happens in the acute noxious stimulation, when a pain then develops into chronic pain state, all these areas get constantly activated into what is known now in the literature as pain matrix. So what happens in the ACC in chronic pain is that increases its spontaneous activity, as it is shown in this paper where they were performing in vivo single unit electrophysiological recording at different time points in a mouse model of chronic pain, where they try to mimic neuropathy by a physical constriction of the sciatic nerve with the cuff. That's why it's called cuff model. What they observe here is that the anterior cingulate cortex neurons from injured animals have significantly higher in vivo spontaneous firing rate, which was also associated with an increase in the general bursting activity, as for action potentials per burst, or in general as a number of bursting cells. Also, ACC hyperactivity coincides not only with mechanical hypersensitivity that is observed in mouse model of chronic pain, but also obviously in patients, as well as anxiodepressive-like behaviors. Here we have previous data from our lab that show that recordings from mice in another mouse model of chronic pain that is called the CCI, and stands for a chronic constriction injury that is also performed at the level of the sciatic nerve, but in a different way. So in these experiments, pyramidal neurons from the layer 5 of the ACC were recorded compared to sham animals. And here we can see clearly that neurons recorded in CCI animals have a lower action potential threshold that matches a higher input resistance. That this means that for these neurons, less synaptic input can lead to a larger voltage change, or in other words, as we can see in the figure B and C, a less current injection is needed in order to fire an action potential, which matches what was found also in the previous paper, but also in the literature, that the ACC displays a high hyperactivity in chronic pain. So as a summary, I hope I can tell you that the ACC is activated by noxious stimuli in both humans and rodents, that is hyperactive in chronic pain, that the neurons in the ACC increase their firing rate differentially during the chronification process, and that particularly the layer 5 of pyramidal neurons of the ACC get hyperexcited in chronic pain. So from here we come then to my research question, that is, what is the role of this output population in the modulation of pain? As we saw in the descending, in the top-down circuit, we understood that the ACC has quite an important role in this. So I want to understand what are the neurons of the layer 5 that contribute to pain processing. And why I want to understand this is because our hypothesis is that specific subpopulation within this layer 5 can undergo different plasticity mechanisms in chronic pain. Because, you know, with experiments of, for example, imaging, we see that there is an hyperactivation throughout the entire cortex of the ACC across the all layers. For me, it's difficult to believe that there is no cellular specificity in that. So to answer my questions, I first characterized the pyramidal neuron subtypes that constitute layer 5. Then I validated the chronic pain mouse model and identified the principal neurons that indeed modify their intrinsic properties in a pain condition. So to know which principal neurons are in the layer 5 of the ACC, we proceeded with the morphological and electrophysiological characterization. So from the literature, what we know is that there is in the cortex a proportion of neurons that enter the corpus callosum and that project within the cortex to other cortical areas, and that these neurons are characterized by a slender apical dendrite, whereas other pyramidal neurons instead can enter the pyramidal tract and go further down to the spinal cord, and these neurons instead have a thick-tufted apical dendrite. So these two groups of neurons clearly have a different morphology that I summarized here in these two reconstructions in different colors, but we also measured several parameters, like, for example, the location within the layer 5, meaning how deep they are in the cortex, the area of the soma, the section of the apical dendrite, the number of oblique dendrites that we can easily count from the reconstruction, and also a quantification of the arborization expansion, considering the complexity of both apical and basal dendrites, meaning pretty much how much area is covered by the arborizations of these two neuronal types. Then this heterogeneity that we observe in the morphology is also clear in the intrinsic electrophysiological properties. So on the bottom left here, we can appreciate these recording traces in response to the same steps of current injection from negative to more positive depolarizing values. So for example, what we can see here is that in response to a negative current, the green trace has a little voltage drop that is called sag, and this is due to the presence of ion channels that only thick-tufted neurons possess. Also, if we move to the depolarizing current injection, we can see that, for example, the firing pattern is also different, because we see, let's say, only by the outlook, that the action potential look different. But besides the look, we also quantified all these passive properties that I've been trying to briefly explain. And we observed significant differences in the input resistance and time constant, which reflect differences in the size of the cells, but also different ion channel composition of the excitable membranes. Then I quantified the sag ratio and the sag amplitude. This DAP, which stands for depolarization after potential, that is this little shoulder right after a spike that is due to other sodium or calcium channels that are mainly possessed by these thick-tufted pyramidal neurons, and also different Reo bus or action potential threshold. So what I did afterwards was to, after I characterized all these morphological parameters and electrophysiological features that are named here as predictors and are enlisted based on the importance, I wanted to, let's say, remove my bias for this classification, because so far I was recording neurons, reconstructing, and then characterize the morphology. So it was me and the neurons. And what I wanted to do is to remove my subject opinion on that, on the classification. So I fed all these parameters to an algorithm and use that dimensionality reduction approach. And what I have is two clusters of, so two neuronal populations, even if we can see some overlapping neurons or neurons that are in between that are, let's say, mistakenly assigned to the incorrect group. However, I would say that let's say all the population of the neurons are not all 100% belonging to one class or another one, because some can have a slightly different morphology or, you know, features that are comparable between the two groups. However, the probability of assigning the neurons to the correct group with this algorithm has an accuracy level of over 80%. So once we have established that there are two populations of pyramidal neurons, we wanted to check how they are involved in a chronic pain model. So what happens? Okay. So in my experiments, I have used a model of inflammatory pain using the complete Freund's adjuvant or CFA that can be injected in the paw of the mice, and this triggers a strong immune response, and is generally used to mimic persistent inflammatory pain, and a saline injection instead as a control. We decided to observe the effect of the acute inflammation at day 1 after injection. And for the chronic phase at day 7, I have two groups. One group of mice that has been left in pain due to the CFA injection solely, and the group instead that has additionally received a daily noxious stimulation, mimicking the stimulus-evoked pain condition. To check whether the CFA mice developed hyperalgesia, we performed the Fomfrey test that consists of poking the paw with the filament until we observed a withdrawal of the paw. And this mechanism, this machine also measured the force in grams at which the mice respond. We can see indeed that in the 3 groups of injured mice at day 1 and at day 7, the mechanical threshold is lower than the control mice, meaning that they withdraw the paw to a weaker pressure. And this means that they do indeed develop mechanical hypersensitivity, and that is long-lasting up to day 7. They also develop thermal hyperalgesia, as we measured with the hot plate test on the left, where we place basically the mice on a metal plate that is set at 53 degrees, and we measure the latency to the paw withdrawal as well. And we can see that this is pretty much consistent between the 3 groups of animals. Then we also used a tail immersion test, where instead we immerse a small part of the tail in hot water, also set at 53 degrees until it flicks. And this test is generally done to see if the pain that is derived from a local inflammation that is in this case at the paw of the animals can also spread into a more generalized hypersensitivity. And indeed, we observe a significant decrease in the latency only at day 7 after CFA injection. It was not present in the acute phase of the inflammation. Afterwards, we recorded passive properties from these animals at different time points, and we observed that at day 1, subcortical neurons that have a SAG and that are recorded from injured animals show a low input resistance and time constant and a higher rheobuzz. So they appear to be instead hypoactive compared to the neurons recorded from the control mice, as we can see also in the frequency of the action potential. We also observed some significant differences in the anatomy of the action potential, because the ones recorded from CFA mice are broader in terms of half width with a smaller amplitude. So what was known in the literature as the ACC being hyperactive in chronic pain states might be not entirely true during the chronification of pain. So as we can see here in the acute phase, we have instead a complete opposite phenotype. So at day 7, instead, subcortical neurons from injured animals show instead no difference compared to the control mice. So there is no difference into the input resistance, membrane time constant, or rheobuzz. So the firing rate of these two between these two groups is completely comparable, meaning they let's say the excitability of these neurons is not altered at day 7 if the mice are simply left in pain. Instead, in this other slide, okay, in the group of mice that were injected with CFA, but that they also received additionally a daily noxious stimulation with a pinprick needle, we finally saw this phenotype of hyperactivity that is typical for chronic pain condition. In fact, we have a higher input resistance and a higher membrane time constant that matches, in this case, a lower rheobuzz, a lower action potential threshold, meaning these neurons need definitely less current injection in order to spike. There was no difference in this case in the shape of the action potential, but what we want to point out is that we did observe this hyperactivity only in the stimulus evoked pain condition, which makes it very difficult to study spontaneous pain that is typical of the chronic pain patients. I'm sorry, I didn't click enough. So in this last slide, I wanted to mention that I didn't notice any change in the excitability of neurons with the slender apical dendrites that project to other cortical areas. So I couldn't find any difference whatsoever in any of the time points or in any of the conditions. So these neurons do not seem to participate into this kind of hyperactivity phenotype. As conclusions, I can say that I found these two populations of neurons in the layer 5 of anterior cingulate cortex that are differently involved in inflammatory pain, that only layer 5 subcortical neurons change their intrinsic properties in inflammatory pain, and that they do increase their firing rate only in presence of a noxious stimulus. Yeah, and I am at the conclusion, I think. Yeah.
Perfect. Thanks a lot.
I guess
Awesome.
I was a bit too early.
No, that's fine. We still have, it means that we have some time for questions if anybody in the audience wish to ask some.
Hi.
Hi.
Yeah. Okay. I have a questions in line with you have asked me. You know what's the role of NGF in pain? I mean, it's related with inflammation, or what have you been studying with this?
So my studies with the NGF were in my master thesis, but I remember very well that NGF is a pro-inflammatory factor in chronic pain.
Okay.
So that's why I was really wondering about the role that is, trophic and normal for being a growth factor. So being present in the inflammatory milieu, it was constantly activating these the nociceptors that are the first sensors of painful stimuli that are at the periphery. So the activation of these nociceptors constantly activates the dorsal horn of the spinal cord, and therefore every time the information goes up. And since in chronic pain, this inflammation is always present and is a marker of all chronic pain conditions. In case you have a neuropathy or in case you have simply back pain, it means that you always have an inflammation ongoing somewhere. Yes. So, and this inflammation is constantly detected, and NGF is part of it. So that's why it's. Okay. I found it funny. Yeah.
Okay. Thank you.
You're welcome.
Do we have any more questions from the audience? Looks like we don't. All right. So I guess that's it for this month. So thanks a lot to all the people in the audience for attending this month's talk. Thanks a lot to Federica and Natalia for the very interesting talks. And yeah, thanks a lot to everyone for their participation. Last chance for the audience to ask some question in case you have some.
For me, maybe I just want to ask Natalia, since I'm now doing, it's completely unrelated, but I'm doing an internship somewhere else and I'm working also on Alzheimer's. So I'll probably contact you for random questions.
Okay.
All right. Some nice networking. It's good to see that. All right. So thanks a lot to everyone and see you for our next NeuroMeetup talk. As usual, you will get all the information either via email through the networks of the University of Bern or directly on our Twitter page. Thanks a lot to everyone and see you next time.
Ciao, Camille. Thank you.
Ciao, ciao. Bye.
Bye.